Hello, my name is Emily Carlson. I'm with the University of Nevada, Reno. I and my co-authors would like to thank you for coming to our talk today. We're going to be discussing the use of ultraviolet light to help control aquatic invasive plants. First, I'd like to acknowledge our funders and give a great shout out to the staff and students who, without their help, this research would not be possible. First, a roadmap of where we're heading. I'm going to start by discussing the background of Lake Tahoe, aquatic invasive macrophytes in general, and the use of UVC. When people think of Lake Tahoe, they tend to think of the world-renowned clarity, the deep alpine lake, and not the increasing population and problem of aquatic invasive macrophytes. Nor do they tend to think of the Tahoe Keys, which is a community built in the 1960s out of a natural alpine wetland that has 2 channels open to the lake. This urban artificial lagoon system can lead to decreased water circulation and increased urban runoff, which in turn leads to warmer water and increased nutrient loading and increased invasive species of plants, which in turn harbor warm water fish, including our friend, the aquarium goldfish. And more importantly to the Keys, the overgrowth of plants leads to reduced boating access. These species present include 3 species of concern, including racoontail. Although it is native, it has become a nuisance, and our usual suspects of invasive aquatic vegetation, Eurasian water milfoil and curly-leaf pondweed. Milfoil was introduced in the 1960s, and curly-leaf pondweed was first seen in 2003. Curly-leaf pondweed is currently outcompeting the milfoil and has become more of a problem, so therefore we are going to focus our research on curly-leaf pondweed. So why are we interested in investigating the use of UVC light? First, it's a non-chemical control method. Chemical methods are generally prohibited in Tahoe, and physical methods are highly regulated. Therefore, we need to look at alternative methods of controlling the plant population. The Tahoe Keys has been using mechanical harvesting for over 40 years, but mechanical harvesting creates a lot of biomass that needs to be disposed of, is also very labor intensive, and it can release viable plant fragments which float to other sections of the lake and cause increased infestation. The boat here was designed and built by Inventive Resources and includes UVC panels that can be lowered into the plant areas and therefore can be used for targeted controls. Our research questions focus on the effects of UVC treatment on native and invasive species at different stages of development, and also, does the results seen in the lab translate to the field? Now moving on to lab experiments, looking at UVC treatment times on various species at different growth stages. Our lab trials were conducted in a temperature and light controlled room using these glass vessels which held about 3 liters of lake water, and the plants were rooted in sand. We treated each glass vessel individually using handheld UVC lights. First, a brief life history of curly-leaf pondweed. They form these specialized buds called turions, which germinate in the fall, in the late fall, and then overwinter as a small sprout. Come spring, they already have a size advantage over other species that are just starting to grow and can therefore outcompete for space. By late spring, they have grown to the surface of the water, and in summer, each plant produces about 30 turions. The turions are shed in late summer and early fall, and each turion is viable for about 10 years, which is why they are such successful invators. So we started our lab trials by treating unsprouted turions with UVC. We saw normal germination and growth, and therefore there was no difference between the treated and control. Thus, UVC has no effect on naturally shed unsprouted turions. So next we treated turions that were allowed to sprout before treatment. What we saw after treatment was an initial decline in the health of the plant. They lost color and turgor pressure. And then we also saw new shoots and new leaves emerging after 3 to 5 days. So as you can see here, the loss of coloration and the new shoots coming in green. Those were allowed to grow for 15 days, and what we witnessed was normal growth. So there were no difference between the control and the UVC. Therefore, recently sprouted turions have enough energy reserves to overcome the effects of treatment. So we decided to retreat the same plants. So here's our first treatment and our second UVC treatment. What we saw was the same effects, loss of turgor pressure followed by new leaves, but at a much slower rate, 7 to 8 days after the second treatment as compared to 3 to 5 days after the first. The turions are starting to deplete their energy reserves. We then saw reduced growth, and after 21 days, they were harvested and compared. The treated plants were never able to fully recover in height. As you can see here, significant difference between the control heights and the UV heights. Whereas after 36 days, the control plant was able to form and shed turions and also shed leaves, which floated to the surface and formed roots. We then treated mature plants. We saw the same initial decline in health, and then a period of resprouting, and then a second treatment was done 9 days later, which we then saw 100% mortality after 20 to 25 days. Some plants had unshed turions. The question is, are these turions viable? We don't think so. When germinating shed turions, we had an 86% germination rate, whereas when we tried to germinate unshed or green turions, we had a 6% germination rate. Therefore, we believe that any unshed turions treated by UVC will be rendered unviable as the plant dies off. We then looked at the effects of various treatment times on different species. We noticed that 2 weeks after treatment, we had only achieved an average of about 50% mortality in the curly-leaf pondweed. So we performed a second treatment on those curly-leaf pondweed plants that were still alive and only those plants. At the end of the experiment, we saw an average of 92% mortality in the racoontail over all the treatment times and an average of 98% mortality in the invasive milfoil averaged over all the time periods. But we were only able to achieve a 62% mortality averaged over those treatment times in the curly leaf pondweed. But we did see the highest mortality of curly leaf pondweed at a 15-minute treatment time. We saw no difference between 20 minutes and the 15-minute treatment times. We then did a community trial where we increased our size and moved from single species to a more densely planted community. These were treated with UVC and then allowed to grow for 22 days. At the end, the plants were mostly brown, but they were not wilted. There were some green sections remaining, and what we assume is happening is that the plants are falling on top of each other and shading and allowing the UVC to miss certain sections of the plant. Now moving outside to the field and looking at a reduction of biomass and the change in community composition caused by the effects of UVC treatment. We performed field trials using the UVC boat to treat all plants in a 1-acre section at least twice a year for 15 minutes, which took about 2 weeks to complete the whole acre. These areas were mixed plant communities dominated by invasives, and unfortunately, the control section became a racoontail monoculture. To assess the efficacy of the UVC treatment, we collected above ground plant biomass before and after treatment. What we saw was a significant decrease in plant biomass. Control is here in gray, orange is the treated, and the periods of UVC exposure are the yellow bars. Side note is that this striped bar here for the control in August is an average of the July and September data because in August the control was so overgrown with the racoontail that it was too hazardous for us to dive. Looking at the treated, we see there is a significant reduction in biomass after the first treatment and an elimination in biomass after the second treatment. It did start to grow back the following month, mostly as newly sprouted turions and racoontail that had moved in starting to fill the void. And 1 year post-treatment without any follow-up treatments, there were no difference in the biomass between the control and the treated sections. We also saw a change in the community composition. In the control, we see this racoontail monoculture with a slight increase in curly-leaf pondweed population year over year. Looking at the treated section, in the same time period, we see a 60% decline in the curly-leaf pondweed year over year. We assume this is because previous treatments have reduced the turion load to the seed bank in this area. Time to break out the popcorn because I have a movie. This shows the treated area before treatment, showing that it's mostly curly-leaf pondweed. And now we have 1 month post-treatment. This is August. We have the control section showing it's mostly racoontail, and then in the treated section, there is no plants remaining except 1 dead milfoil. And then this is 2 months post-treatment, controls on the left again, and then the treated's on the right. Sorry, this is in September, the highest turbidity. The control is that racoontail again, and the treated section shows that there is nothing remaining except rotting vegetation. And now moving on to conclusions, a distillation of the lab and field experiments, and what the future has in store for UVC treatment in Lake Tahoe. So now to bring the lab and the field trials together. We see that there are effects after 3 minutes of treatment, but a 15-minute treatment time is preferable for areas with curly-leaf pondweed. This allows time for the sediment to settle, plants to move, and coverage during any boat drift. Now, areas that are dominated by milfoil or racoontail may require less time to achieve the same results. Plants react up to 14 days after treatment by losing color and falling to the sediment, but they can regenerate at any growth stage. So it's important to treat the plants before the turions are shed, therefore reducing the turion load into the seed bank, reducing next year's curly-leaf pondweed population. Don't really have time to go into the details of the data, but we saw a few other results from other experiments that were pretty interesting. We collected benthic macroinvertebrates pre and post treatment, and we found there was a reduction in the population initially, but overall there was increased recruitment long term. We also compared UVC treatment to mechanical harvesting and saw a similar biomass reduction from 2 UVC treatments when compared to 8 harvests. Thus, UVC treatment requires a lot less man-hours to achieve the same results as mechanical harvesting. And this picture here, you see the UVC was treated on this area, and this is next to the bulkhead where the boat could not reach, and that racoontail is to the surface. And looking at this visually, we saw the best results using a 15-minute UVC treatment in the late spring, which produced an initial decline in plant health, and followed that up with a second 15-minute UVC treatment in summer during a period of high turbidity. The plants then fell to the sediment and were at a low level of sunlight penetration and were not able to overcome the effects of treatment, which then led to increased mortality of the adult plants before the turions were shed, and thus a reduction in next year's population of curly-leaf pondweed. Other research areas where UVC is currently being tested include irrigation canals and marinas, where UVC panels can treat underneath boats and underneath dock slips, recreation areas including sandy beaches, and currently a larger treatment area is being tested in the Tahoe Keys. And who knows, the future might bring us autonomous UVC boats. And lastly, we would like to thank you for the opportunity to present our research findings.